So DNA, the blueprint to life. If we go ahead and look a little closer at the structure, we see that it is a double helix. That I know you know. So it was James Watson and Francis Crick in 1953 that discovered the double helix structure with the repeating units. The repeating units are those nitrogenous bases. There's only 4, and they repeat again and again and again. And the order of these is actually really important. It is the order that determines what type of living thing you are and how your physical features are expressed. So this whole thing, this phosphate, this sugar, and this nitrogenous base is a nucleotide, okay? And each nucleotide of DNA has those 3 things always. The deoxyribose sugar, the phosphate, and the nitrogenous base, which is either adenine, thymine, cytosine, or guanine. And we hold those nitrogenous bases together because this will be a backbone, half of a rung, and then it'll be attached to another half rung and then another backbone. Those 2 rungs or nitrogenous bases are connected using hydrogen bonds. Okay, remember last unit we talked about how hydrogen connected the phosphate, another phosphate to ADP to make ATP. And I told you it was the hydrogen that formed that bond and held it together kind of like a glue. It's doing the same thing in your DNA with each of these nucleotides. So since adenine always likes to pair with thymine and guanine with cytosine, when we look at DNA, there will always be a similar percentage of adenine and thymine and a similar percentage of guanine and cytosine. So there might be different percentages of adenine and guanine, thymine and cytosine, but since adenine and thymine are the pairs, there will always be the same percentage or really, really close to the same percentage of those, and then the same percentage of these. And that is called Chargaff's rule. And that is also because these are different sizes. So adenine and thymine have approximately the same diameter, and so they like to pair up together. The same with guanine and cytosine. They have approximately the same diameter, so they also like to pair up together. Adenine and guanine or adenine and cytosine, the sizes don't match, so they do not pair up. And the same with thymine and guanine or cytosine. So adenine always binds, always with thymine using a hydrogen bond. Think apples go on a tree. Adenine, thymine, A, T. And guanine always binds with cytosine using a hydrogen bond. Cars go in the garage. Cytosine, guanine, C, G. So DNA replication. Now that we know what DNA is made of, we could talk about how it replicates itself. Before a cell divides to create another cell, it has to duplicate its DNA. So it copies its DNA to make sure that each cell will have its own set of DNA. This happens during S phase of the cell cycle. We're going to talk a little bit more about each step of the cell cycle a little bit later this unit. During DNA replication, the DNA molecule will separate into 2 separate strands. And then each strand gets a complementary strand, making a brand new DNA strand. So each strand of the double helix is a template. I take apart the 2 strands of the DNA and I create a new strand on each of those. We'll look at what that looks like. So if this is my DNA double helix, you see they're coming apart, okay? Each strand is coming apart. This one is going from 5' all the way over to 3, and this one goes from 3 over to 5. That's important. So the first step of replication is to separate the DNA. So I've separated these two apart, and I use that using an enzyme called helicase. Remember, we talked about how proteins can be enzymes, and enzymes typically end in ASE. Helicase is a protein or an enzyme. Its job is always just to kind of unzip this DNA. So if you think of a zipper, we have two sides of the teeth in a zipper that we use the actual little part that you handle, that has like a little handle on it, to either zip the strands together or to unzip it. So helicase is kind of like the part of a zipper that you actually hold and drag up or down, or sideways, depending on the direction of your zipper. So helicase unzips the two DNA strands. So where the two strands are separating is called the replication fork. You know, a fork in a road is where the road has two new paths that go in different directions. So the fork in the DNA is where those two strands are coming, and you can go two different directions. Each strand of the double-stranded DNA serves as a template for the production of the complementary strand. What that means is, now I have this side of the DNA with its nitrogenous bases, and it can start building a new strand of DNA onto that using the nitrogenous bases as a template. It knows that wherever it sees an adenine, it's going to pair a thymine. And wherever it sees the guanine, it's going to pair a cytosine. So we talked about how one strand goes from 5' to 3' and the other strand goes from 3' to 5'. Even though they're parallel, they're running in opposite directions. So we actually call that anti-parallel. So one goes from 5 to 3, the other goes from 3 to 5. Now, after the helicase unzips the DNA, we can use DNA polymerase, another enzyme, it ends in ASE, that is going to elongate the new DNA at the replication fork. Remember, that's where they come together or apart. So DNA polymerase is actually responsible for connecting the nucleotides. So it is going to connect the nucleotides onto this template strand, and then it's going to proofread that strand. You have to make sure that you actually did it correctly. You know, the way you guys are supposed to proofread your work before you turn it in. So then ligase will come, and all of these little fragments have spaces because this is running in a different direction. So as the DNA unzips, it will go and fill in that space, and then it has to go back to the beginning and fill in more, okay? So the DNA unzipped this section, and the DNA polymerase replicated that section. And then as it unzipped some more, it had to go back up here and fill in this section, and then stop. And then the DNA strand was unzipped some more, so it goes to the next section and through. The DNA was unzipped some more, it goes to the next section and through. So that actually leaves little gaps where the DNA polymerase was or ended. And it is ligase who comes through and fills in all those gaps. So this is just a closer picture. So this is the leading strand, the strand that just replicates continuously. As the DNA is unzipped, it can just zoom on through and pair all of those nitrogenous bases. But this side, as it is unzipped, it's going to have those spaces that we just talked about from the last slide. So that's called the lagging strand. The leading strand just goes on through. The lagging strand, it takes a little longer because that polymerase has to go this section, and then when it finishes that, it goes back to the front and does this section, and then it goes back and does this section. So it takes a little longer. It is lagging. All right, so here's my helicase. It's going through, it's unzipping my DNA. Here's my DNA polymerase. It is building on new pieces, the adenine, thymine, cytosine, and guanine, and then it's going to proofread that to make sure it paired them correctly. So you see this DNA polymerase is just moving in this direction, and it's going and going and going. And as the helicase is in front of it, unzipping, it just keeps going. Whereas on this side, the helicase has unzipped, and so it's going to start here and move its way down. And when it gets to the end, the helicase will have unzipped some more. So it goes back to where it's now unzipped and fills that in. And it has to keep doing that over and over again because it has to work in a specific direction. It can't work in this direction. So if I go back to the slide, it cannot work from 5' to 3'. It has to go from 3 to 5. Okay, we start lower at 3 and work our way up to 5. So the genetic code. This is a universal code. This says there is a single evolutionary history that underlies the origin of all organisms on Earth. So the sequence of our nitrogenous base is important for the code for genes of a particular species. So remember I said earlier that the order or the sequence of those nitrogenous bases determines what type of organism you are. And that's all it's saying here. If we change the sequence, that can be inherited possibly by future generations. So it can actually change the species. So things to remember, which nitrogenous bases go with which. So you should be filling this in with adenine, guanine, thymine, cytosine. Which ones go with which? And then I want you to write which goes with each of these. So if I were making another replicated piece of DNA here, what would you pair with this cytosine? What would you write across from it? What would you pair with this cytosine? What would you pair with this thymine? And this adenine, and thymine, and adenine, and thymine, and cytosine, and all the way down. So you should have, however many letters this is, you should have the same number of letters on this side of the complementary bases. And that's it.